This video will show how to display graphically a pedigree of genetic lines that are created during a typical breeding program, including all traits measured on them. In the Analysis page, I'll go to the Studies filter and then select the Program A folder. In this database, traits names are coded like T1, T2, or P1, P2, and so on. Next, I'll pick the Phenotype Overlay option under Pedigree. Now I can put one or more traits in the Y field to be plotted on the pedigree. Let's start from a single trait and put P1 into the field. Then I'll check Show Labels and click OK. And this is the pedigree plot. Here, each symbol represents a genetic line or germplasm. Symbols above it represent its parents, and below are its offspring. The symbol's color is the trait's value. The color's legend appears on the top. Going back to the From tab, it's possible to put several traits in the Y field to represent all of them. Let's add these traits and see how it affects the results. Now, instead of a circle, we get a box that's comprised of colored squares. A box represents a genetic line, and each square is one of its traits. This legend shows the location of each trait within the box. On the top, we have a legend for each trait's values separately. In this case, they are all the same. So the figure allows following a trait's inheritance throughout the generations. I can also select items from the figure. Clicking on an item will add it to the list below, just like this. Then I can click here, and the lines are displayed as a table, and I can drag them into the lists from the left. Continuous traits can also be displayed on the pedigree. Let's select T1. And this is how it looks like. Now colors represent continuous scale. Green represents low value, black is average, and red is high. Of course, I can also take several continuous traits. Let's take this one. And again, each symbol appears now as a box, with each square representing a different continuous trait. The next example I'll take from the tomato database. Here I'll go to Tools and then Manage Tool. On the left there are germplasm sets, which are lists of the genetic lines. Double-clicking on a list will display all lines in it on the right. Then I can select a subset of the lines, or even all of them. And then click on this button. This will present selected lines along with their pedigrees in the open source Citoscape application. This is how the pedigree looks in the Citoscape application. I can zoom in and out using the mouse scroll, and then adjust the zoom window and walk with it through the pedigree. Orange edges indicate that these genotypes were derived by selection. Pink and blue edges represent crosses. Pink connects a node to its female parent, and blue to its male. When I click on a node, it is selected and highlighted in yellow. Its traits data appear below. I can select several nodes by opening a rectangle around them and observe the trait values. A node can be dragged to view its connections. As another option, I can color nodes according to a particular trait. For example, let's take the trait firmness. This shows the possible values of this trait, and each value is assigned a color. Then, all nodes of the pedigree are colored according to their trait's value. For instance, this purple node has firmness of 3, as can be seen here and here. This one is 4. I can change the trait that will be used for coloring. For example, let's pick this one. Now nodes are colored by the fruit locule trait, and we can change it to any other trait. And this is fruit size, and this one is inflorescence type, and this is the trait fruit color.